Okay, so while I'm putting it on live, could others know, tell me about their background as well? Pradeep says he is from Periyar University, India, Tamil Nadu. Good to have you here. Okay, so we would be live on YouTube, and if anyone misses out on anything, they can just join on YouTube as well. I'll be sharing the link with you all. Hi, Pradeep. If any one of you has any question in the middle of the session, you can then and there put it in the chat box, and our team would be there to answer your question. I hope everyone has joined, and now let's begin with the webinar, while others can join in the midst of it as well. So a very warm welcome, everyone. Thank you for joining today's session. And I would like to welcome you all for today's webinar on the overview of OmicsLogic Genomics in the virtual lab. Today, we would start with the introduction to the OmicsLogic training and platforms, and then we would hear from our experts about genomics and future medicine, followed by a video presentation by our program mentor, Dr. Sona Vasudevan, and then we will go through the schedule of the entire program and go through the upcoming opportunities as well. So before we start with the talk, I would want to introduce us. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. OmicsLogic is an international program that is running in 5 different regions with over 10,000 users around the world. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and leverage our online training resources, adapting them to the needs of the students and researchers around the world. OmicsLogic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. My name is Sonalika Ray, and I'm the OmicsLogic Community Manager responsible for daily interaction with thousands of users that are a part of a bioinformatics community managed by Pine Biotech. Now, coming back to the program, Genomics in the Virtual Lab is a special opportunity to learn about genomics and big data analysis from world-renowned scientists and a team of bioinformaticians working on cutting-edge big data challenges in biomedical research. In this online program, participants will learn about the analysis of high-throughput genomics data and would apply complex and computationally intensive data processing steps to large-scale public domain data. Now, let me introduce our program mentors and guest speaker. We have Dr. Harpreet with us. She has an expertise in the application of statistical machine learning techniques on biomedical data and the development of prediction tools and databases. And she looks after the curriculum development, bioinformatics and data science course development. Then we have Dr. Mohit Mazumdar. He is an experienced bioinformatician with multiple publications in peer-reviewed journals and collaborations in industry and academia. And over the past several years, he has been developing collaborations with universities as a mentor and project lead for OmicsLogic Training and Research Fellowship. Ilya Brodsky is the CEO of Pine Biotech. And then we have our program mentor. She is Dr. Sona Vasudevan, Professor of Biochemistry and Molecular Biology at Georgetown University Medical College. She would be mentoring our students and would present disease-associated projects that our participants can learn from and ultimately see how genomics is used at the intersection of data science and biomedicine. Dr. Arigi, Research Associate Professor, Center of Bioinformatics and Computational Biology, Department of Computer Information Sciences, University of Delaware, would be our guest speaker for the program. Dr. Arigi leads the UniProt database efforts and leads the biocreative and biocuration efforts as well. Now, I would like to pass it on to Ilya, who will be the co-mentor of the program. Over to you, Ilya.
Thank you very much, Sonalika. I'm going to share my screen. Okay, so welcome everyone to this webinar. It's exciting to see all of you here today. And what I will try to do in my part of the presentation is to briefly tell you about the reason why this program is the right place for you to start as you're learning about genomics. So first of all, let me briefly introduce myself. I have been working with PHYLOGENY Biotech for several years now, and I have been a part of many different presentations and training sessions that were related to bioinformatics for a variety of different levels, starting from faculty and graduate students, but also including high school students. So we have built a team of academic advisors and collaborators, as well as facilitators that will include many different types of people that can help you along the way. And what we have really been focused on is how we can use our expertise and the network that we have to bring to you the best possible experience learning about bioinformatics in a virtual setting. So how do we take what we have successfully done offline and bring it online in a way that all of you could participate and get an equally excellent experience learning about applications of big data to a variety of biomedical challenges, but also how to work on translational research challenges that are relevant to where you are right now. So let me start by kind of telling you why bioinformatics and genomics is such a relevant topic today. So from the 1950s, after the DNA double helix structure was discovered, right away there was a focus on trying to understand what is encoded in the DNA. And so as a result of the Human Genome Project, and that led to many other species being sequenced, we started to really accumulate a huge volume of data. And that volume of data today is accessible to anyone that has access to the internet. Now, what's interesting about this is that this data actually comes with a lot of variation. And so the topic of genomics, the study of genomics, is really focused on looking at the DNA code, trying to find patterns and bugs in the code that might lead to phenotypic changes or changes in the actual functional molecules that our cells use to operate and function as we grow. And so these bugs in the code could result in major changes that are associated with a few different important biomedical challenges. And these include drug metabolism. So for example, the function of different enzymes that process the drugs that are prescribed to patients. And that could result in patients being non-responsive to treatment, or maybe even having a toxic reaction to a treatment. That has to do with cancer. So a lot of cancers are associated with what's called somatic mutations, mutations that accumulate as the cells divide. And as they accumulate, that might disrupt some of the regulatory networks that control different properties of cells and make them into what they are, causing tumors to grow and causing them to spread or metastasize. That has to do with inherited diseases, and those inherited diseases could be a result of many different environmental factors over a long period of time, as well as rare genetic diseases. And so those rare genetic diseases could be something that essentially endangers the person at a certain time or prevents them from developing certain important functions as they grow. And so all of this is linked to a variety of different types of genomic information that we could acquire. And what's interesting is that as opposed to just maybe a few years ago, this is now in the clinic. So genomic medicine and the application of genomics in treating patients, interacting with patients today is not just something that we talk about happening in the future. This is happening right now. And it starts from collecting information that would help us interpret the found genomic mutations in a patient sample so that that could be incorporated into the treatment plan. So today, this is already being used. There are many different genetic tests for history, for different kind of predisposition of risk, but also for treatment and for accurate diagnostics. As I mentioned, in cancer, this is a very critical component of cancer care, including the identification of early stage signatures that could help us treat cancer better. So as you know, cancer is a huge challenge around the world. In many different parts of the world, there are different types of cancers: liver cancer, breast cancer, lung cancer. They kill millions of people every year. And what is a critical step in being able to address cancer better is early detection. So the earlier we can identify a cancer developing in some cells, the earlier they could be treated. Another important part is accurate and personalized treatment. And so that has to do with genomic information as well. As you all know, a good example of that is the p53 gene. This is a gene that is typically found in many of the tumors, in many different types of cancers. And there, just a small number of mutations can cause that gene to stop functioning. The protein that is encoding will stop functioning properly and essentially will lead to unproliferated growth and uncontrollable tumor development. So this is an example in cancer. As you know, as I mentioned, there are other types of genomic information associated with disease, for example, cystic fibrosis. And to really appreciate the diversity of information that genomic data provides, it really could be applied in so many different cases. And there's a complex way to kind of categorize these types of mutations to really understand what is their impact. And so you can see that here we can see a number of different types of mutations that are affecting the protein synthesis, the maturation and processing, ion channel gating, and others. And so when we look at the data that is generated about the genome, we really need to know how to analyze this data, looking at single point mutations, and also sometimes looking at whole regions that are being changed. And so as a result, when we speak about genomic data and the variety of different use cases, we can apply genomic surveillance, genomic analysis for variant calling, as well as unsupervised analysis of genomic data, ultimately to find associations between the genomic variants and the phenotype. This phenotype could be disease, as it is caused by what's happening in the human DNA, in the host DNA, in the patients, or it could be caused by pathogens. And so another whole important area of application for genomic data is the study of genomics from pathogens, from viruses, bacteria, and from parasites. And so tuberculosis is one of those examples. As we know, there is a huge problem with drug-resistant bacteria that is now not responding to a single type of antibiotic, or what's called a multi-drug resistant bacteria as well. And those could be detected by analyzing publicly available genomic data. So another example for this kind of application is the Ebola virus. As we know, in the 2014 to 2016, the outbreak of Ebola in West Africa caused the whole world to be concerned about Ebola spreading around the world and causing a pandemic. But thankfully, because so much effort has been put into addressing this problem early on, the understanding of what causes the mutations that are being acquired in this new type of Ebola strain were identified. it was identified what is the critical protein, and a vaccine could be designed very quickly. And so that was a combination of factors. On one hand, genomic data availability and the efforts of researchers around the world. On another hand, it's the understanding about the relationship between sequence and structure, which is a very important part of what we will be talking about in this program. So one side, it's the genomic data, as you can see right here on the right. On the other side, it's the proteins that are being encoded by these specific genes, and how do these mutations actually fit into that picture, and how we can utilize that. So as I mentioned, at the foundation here, we have the sequencing technologies that have over the years diversified, providing us with a variety of different types of sequence information. And to understand how to process this data, we have to understand how to analyze this data properly. And so that's why in this program, we'll talk a little bit about the history of genomic data sequencing, how to analyze and identify specific mutations, how to categorize these mutations. And so there's a lot of individual types of mutations that we could think about, but we will try to go through all of the different types and terminology so that all of you are prepared to look at genomic data and actually find associations with a phenotype that you could link because you understand the data. And so that brings me to this field of bioinformatics. So ultimately, what we talked about up until now is the application of genomics in a variety of fields, primarily in biomedicine. But at the core of being able to perform such projects and learn from this data lies this bioinformatics discipline. And the bioinformatics discipline combines the understanding of biological challenges or biomedical challenges that we just heard about with two important aspects, informatics and statistics. And so informatics has to do with information technologies and data management, how to find data, how to download data, how to annotate this data, how to store this data in certain formats. And then statistics, how do we analyze the amount of data that we have to get some meaningful information out of it so that we can do inference, so that we can infer from this data associations with meaningful patterns that we know about the patients that we are studying. And so the intersection of these three fields, informatics, statistics, and biology, is actually the definition of bioinformatics. And so to talk about bioinformatics and to help people acquire skills in bioinformatics, I want to introduce you today to the OmicsLogic portal and some important things that you can gain from this portal as we speak about genomics in the virtual lab, the program that I will show you in just a brief moment. So if you could please go with me to OmicsLogic, the portal. So maybe somebody can share the link. Okay. So the link is learn.omicslogic.com. Thank you, Sonalika. Please go to this portal. And if you could please register on this portal, this is free registration. And anyone who is participating in the program or who would like to participate in the program would be leveraging some of the resources we have on the portal to be able to go through the session. So I will give you just a couple of minutes. If you could please register on this portal, the registration is free. And then we will speak about the topics and the process for actual registration to the program. Okay, so once you register, please put number 1 or yes in the chat so I know that you're already registered. Okay, thank you. I'll just give it a couple more minutes. So all of you that are registering, as you register, please let us know in the chat. And then I'll be able to show you a few more things so that everyone will be able to appreciate what kind of resources are going to be available for you in this program. All right, great. Thank you. I see a few of you are registering right now. Okay, so once you have registered, you can go ahead and navigate to this tab called Courses. And I'll just give you a few examples of what has been made available through this portal and how it fits in with what we're going to be doing. Okay, so once you're on the Courses tab, you can go ahead and navigate right here, if you're in the Search tab, to a course called Bytes and Molecules. Now, this is a free course. Anyone can take it, and this will help you establish some of the terminology and the basic concepts behind genomic data analysis. It will talk about why we should study genomics, how we should study genomics, and what are some of the exciting examples of the application of genomics in a variety of fields. So you can learn about this by completing this course. Another course that I would recommend for all of you that are joining today is a course called Introduction to Bioinformatics. So the first course will give you the general background and the terminology, and will refresh your molecular biology concept so that you understand how all of this works together. Introduction to Bioinformatics will introduce you to the challenges and opportunities with big data, such as next generation sequencing, and how this data could be analyzed. And it will go on into a more detailed description of the applications of bioinformatics in the field of biomedical research, clinical applications, pharmaceutical drug discovery, as well as biotechnology and agriculture. So I highly recommend for all of you that are interested to join the program to start looking at this coursework. It's very accessible, it's free, and you can use this right away to prepare for the program that we're going to be starting. Now, to talk about the program, we can go on the main menu right here to the Programs tab. And on here, you will find a variety of different training programs that are not asynchronous. They're not online courses. They are guided by a mentor. And so this program called Genomics in the Virtual Lab is going to be found right here. And in this program, this is going to be a special opportunity because it is going to be guided by someone who has years of experience working in application of bioinformatics to biomedicine, but also a program director that is very passionate about education. And so instead of teaching her master's degree program over the summer, she's actually going to be spending her time working with students and beginners to give them the opportunity to appreciate bioinformatics and genomics in the context of biomedical research. So I think this is a very exciting opportunity. She has years of experience working for NIH, working at Georgetown University Medical School, where she is participating in many different exciting research projects. But also she is a director of a program called Systems Medicine, and really a leader in the field of systems medicine, where they speak about the need to integrate data, including genomic data, to be able to offer better treatments to patients with a variety of different diseases. So to understand what the program is going to be like, I think we here have a video of a group project that the previous participants put together. You can scroll down and see some information about the schedule. And the important thing is that the time for the program is going to start from the 21st. So on Monday, we will have our first session, and it will go on for 2 weeks. And during those 2 weeks, you'll be able to learn what you need to know to get started with genomic data analysis. But in the later part of this session, we'll talk about some of the other outcomes for this program as well. So before we continue, we want to mention what the program is going to be like. And so I think we have Dr. Sona Vasudevan from Georgetown University, who's going to speak a little bit about the history of bioinformatics and how it relates to Georgetown University, really the school that she represents. So you can hear a little bit about the connection between bioinformatics and genomics for Thank you. Okay, go ahead.
Now, there are lots of subdisciplines within bioinformatics because bioinformatics is an interdisciplinary field because we need computer scientists, we need biologists, we need even economists right now, right? So it's an interdisciplinary field. So there is a development of new algorithms and you have statistics because the minute you talk about big data, you need statistics because you want to see if this one single mutation is actually statistically significant for us to say, yes, this particular mutation causes that particular disease, right? And then we have analysis and interpretation of various types of data, including nucleotides, amino acid sequences, protein domains, and protein structures, and how do we get structures to function, and all of those things. And then the development and implementation of tools, because we can generate data. We can have computer scientists to develop tools to look at the data. And we also need, without tools, we cannot even mine and look at this data. That's what this course, you're going to be learning about a lot of resources where there is data, but also you're going to be using tools to be able to use, how do you use the data without tools, right? But we are going to be focusing just on this aspect of nucleotides, amino acid structures, and all of that for the first 1 week, right? Now, is informatics a new field? What do you think? I really love to ask this question to all my students, and I get different kinds of answers. So I would like to know, of course, I wish I had some polling set up so that we could have just polled. Now, what do you all think? Is bioinformatics a new field or not? You can just type yes or no answer into the chat, and we'll see. We'll just wait for a second and see. Okay, no, bioinformatics is not a new field. Okay, I'm seeing a lot of new, no. That is actually, no, okay. So bioinformatics is actually, you're right, is not a new field, right? So where do you think it was born? So bioinformatics is not a new field, but where was bioinformatics actually born? Who gave to this whole field of bioinformatics? Any guesses of which continent? When I went to school, it was 7 continents. I don't know how many continents we have today. But how do you, Rosalind? So you mean Rosalind Franklin. Okay, where was it born? Which continent was it born, you think? Any guesses of where bioinformatics was actually born? Asia, okay, that would be my guess as well. X-ray crystallography, chromatography of DNA. USA, okay, probably in Europe. Okay, so we're covering a lot of different continents here. We've covered Asia, you've covered Europe. Asia is coming up a lot. Europe is coming up a lot. UK, yeah, sure, okay. So Cuba, okay, good guess. Actually, I'm going to take someone who just actually, maybe one who made the protein atlas. Okay, we are actually coming really, really close. That is very true. So bioinformatics, I spent a lot of time on the slide just because it's really fun to see how people think, right, where it was born. Yes, it was actually, I'm going to get closer to, I think it was Parimala, or someone else said USA, right? So I think whoever said USA, it is actually correct. So it was actually born in the US. And it was actually, I'm very proud to say that it was actually born in the universities I work in, Georgetown University. And I think Parimala, you're actually right. It was actually the person who actually made the protein atlas. And who is that person who made the protein atlas? Any guesses? So before I introduce, I have to give tribute to this person, Robert Ledley. He was a faculty at Georgetown. He actually died from Alzheimer's disease. And he was the person who actually discovered the whole body CT scanner as well. And he actually published a paper in 1959 where he said, he predicted that 50 years or 30, 40 years from when he wrote that paper, that we are going to be drowned in data and we are going to be dealing with biological data. He is the person who actually hired the person to Georgetown who actually gave birth to the entire field of bioinformatics. And she is actually officially now called the father and the mother of bioinformatics. She was declared the father and mother of bioinformatics by David Lippman, who actually was the director of NCBI, which we will talk about. So yes, those of you who said it's a lady, yes, it is a lady. And she was the one, and she actually came up with this atlas of protein sequence and structures. It is actually before my time as well, in 1965, right? So this is, so in 1965, I don't even know how many years ago that was, where people used to actually get sequences published in a book. People used to sequence and send the sequence to Margaret Dayhoff, and she used to just publish this in a book. And this book was the one that was actually posted to all the scientists around the world. And that is how they used to get the protein sequences, right? So now, what were her other seminal contributions? Yes, someone said, I think it was Parimala who said, Protein Atlas. And she gave birth at that point. She was a visionary. She's considered a visionary because people who are visionaries, they already think what's coming 50 years down the road. She was the one who said, well, we are going to have a data explosion. So let's create what is called a database. So she was the person who first created the protein database. And if any of you are visiting Washington, D.C. sometime and you visit Georgetown, I can take you to actually the servers and all the books that she published. We still have it because, you know, I used to hate history in school, but now I love history. And so I have actually kept all her papers and everything else as everything was coming along. And she also came up with a single letter code for amino acids. I'm sure you all know what single letter amino acid letter code is, right? I'm sure. I don't want to quiz you on that. But is there any reason, do you think, that she came up with the single letter amino acid code? For example, glycine, GLY is G, right? Alanine, ALA is A, right? So do you know why? Yeah, efficiency. But other than the efficiency, of course, it's very efficient, right? Today we look at it and it's extremely efficient. But think in 1960s, why would she come up with a single letter amino acid letter code? What do you all think? Easier representation? Yes, that is definitely true. But other than that, you know, you all are in a generation where we have gigabytes of data in your phone. You're carrying gigabytes of data in your cell phone. So you can do this whole thing in a cell phone, right? Back then, yeah, it was lack of computation power. Back then, they were talking about kilobytes of data or even megabytes of data. So they did not have the supercomputers. So they wanted to make sure that they don't have enough space. So they had to actually converted that to single letter amino acid code. In fact, I learned all these stories from people who worked with her, who are actually still at Georgetown, right? Coding, storing information is efficient. That is correct. So now from that, where did we head, right? 1950s. And of course, you all know about Watson and Crick, right? Without Watson and Crick, we won't be here today. And then we have Sanger sequencing. Without sequencer, we could not have sequenced anything, right? So insulin was the first protein. And then, of course, things started evolving. We started seeing a lot and lot more. Excuse me. We started seeing, and then we started seeing polymerase chain reaction. And then, and then we started seeing NCBI, and then we started seeing all of these different databases. And it was in 1991, the official term bioinformatics was coined, and it came up in the literature. Excuse me, I think I'm screaming a lot here out of excitement because of all of you, right? So then we started seeing all of these tools that was developed and all of those things. And then we started developing, and a lot of different things started. And you can actually pay attention to here where the whole genome project initiative was actually started in the 80s. Excuse me. And then the origin of bioinformatics, the first protein sequence reported was that of insulin, 1956. It consisted of only 51 amino acids. And then nearly a decade later, the first nucleic acid sequence was reported. Again, there is a faculty at Georgetown who actually passed away 2 years ago, Jack Chirikian. He was the first person to actually sequence a tRNA when he was at Princeton. So
Thank you so much, Ojasvi, for sharing the recording with us. So that was Dr. Sona Vasudevan, who told us about the brief history of bioinformatics and discussed what the bioinformatics plays a role and how it does so. And she would be our program mentor. So before I pass it on to Dr. Mohit to discuss the session details, I would quickly want all of you to please answer few of my questions in the poll. Maybe I can take the poll after Dr. Mohit as I see some technical glitch over here. Over to you, Dr. Mohit. Thank you.
Hello, everyone. Just a second. And thank you, Dr. Sona, for that wonderful talk. And before that, Ilya describing genomics. And what I wanted to discuss with all of you, can you see me now? Yeah, hello,
Yes, we
everyone.
can.
It's great to have you all here and really happy to be in this session. So today, as you have already learned about genomics and application of genomics and why bioinformatics and how bioinformatics started, now I'd like to tell you more about the program itself. So this is a great opportunity, genomics in the virtual lab, especially for those who are starting with this field, who are new to this field, and especially designed for high school students and beginners. So in this program, we will have different phases. And before I tell you more about the program, let me tell you a little bit about myself. So my name is Mohit, and I am a biomedical data scientist. So my specializations are in NGS data analysis, and I am proficient in applying machine learning and has detailed experience of working on structural biology and doing molecular dynamic simulations and doing modeling. So in this program, I'll be one of the co-project mentors along with Dr. Albrecht. So one of my research interests and what am I doing currently is that about the structure function thing that Ilya talked about. So currently, we are working with a group in Ghana, and in there we are working with infectious diseases such as malaria and COVID-19, which is a very big issue, especially malaria in Ghana. So in the project, we have been able to look into the mutations that could be, that could help design vaccine strategies. So we are using genomics, and for me, I'm doing some of the structural analysis, helping connect that how mutations actually influence the function of the structure. So how mutations changes and why these mutations are important and why some of the mutations are not so important, which are to focus upon, and how we can develop strategies like vaccine, how we can develop strategies like small molecules that can actually go and block these deadly disease to spread or to stop the severity of these diseases. So before that, I've been working with several labs and working with several students. So during my PhD time, I was able to teach a group of students who are doing their MSc in bioinformatics, and then since I joined Fine Biotech, I've been able to communicate with so many of great minds here, students from high school and scientists, and also people who are doing their postdoctoral studies, PhD projects. So it has been really amazing to be able to work on several of these important aspects, being a person who's been able to understand what data is. So to be able to work on sequence data, to be able to work on structural data, we have designed this amazing program. And now I would like to go next to tell you that how this program balances out both sequence, introduction to bioinformatics, and then take you to the next level of application of bioinformatics. So in this program, we will have different stages. So there will be week 1, there will be week 2, and there will be week 3 and week 4. So this is a 1-month program, as you can see. And during this entire 1-month journey, we will start with very basics of introduction to bioinformatics, which is very important. And also Ilya talked about the course. So please look at that. So it will start with detailed video session by Dr. Sona. That would be the first day, June 21st. And then we will have different aspects of how bioinformatics is applied, and we will go into protein structures. We will go into 3-dimensional structures from sequence to understand how these relationships are there to understand and annotate things. So in bioinformatics, what happens is that we do a lot of comparison, and we will use multiple sequence alignment and phylogenetic analysis to do such comparisons and understand those relationships. And then, as I was telling you, that the exciting part of is to able to come up with the holistic understanding from sequence level to a structural level to a functional level. And then there will be a guest lecture, which I'm really, really excited about. And then we have the second week where we'll be discussing about detailed developing a project in the context of application of genomics. So in this entire program, we will be using several cloud-based tools and tools that you can download and apply in your own systems as well. So there will be a lot of computational tools that you will be learning about a lot. You'll be learning about a lot of algorithms and how they work, and you will be also able to do hands-on practice on all of them. We have also prepared data sets for you to take and also learn and apply this. So we have already compiled genetic data sets which have sickle cell anemia, cystic fibrosis, and all of those diseases. Or also you can work on your own data set. So if you have been thinking about a disease that you want to work on, if you have that interest, we can obviously look for data sets in this entire program, and then we will be able to work on the project. So now I'll just quickly take you to each of the sessions that will happen during this entire course. And also then I'll take you to, I'll tell you that what you will learn from this. So the first session will be about introduction to bioinformatics, as you can see on the screen. And in this session, we will be working on, we will be learning about omics technology and get an overview of databases and resources that is available to carry out data-driven research in sciences. So in this session, we will learn about the introduction to bioinformatics and different fields that are there and applications. In the second session that I was telling you about, that we will be learning about genomic variants. So in this session, we will learn about DNA, nucleotides, and amino acid variants, as well as how this structure and function correlate. So what happens in structure, how it affects its function. So think about it like that. If something changes, so in the context of protein, how this works is that from amino acids or from nucleotides, we come to a level of amino acids, that is through transcription and translation, right? And then we have a protein which is formed from 20 amino acids. So you have a lot of amino acids in a chain that forms a protein. And then when you study here in this session, you will be able to what? You will be able to visualize this, that how in a 3-dimensional space, the protein how it looks, what are the residues it is built of. You'll be able to look at the chemistry. So you'll be able to look at the nucleotides that are there. You'll be able to look at the individual charges that are there. Like if the amino acid is built from carbon, nitrogen, oxygen, what are the bonding, all of that. So you will be able to visualize and learn from the aspect of protein structure and function in the second session. So after this session, the third session will be about sequence alignment. So in this session, we will learn about multiple sequence alignment of both DNA and protein sequences. And this will be again the hands-on session 2 will be about mutations and in 3D structures. So as I was telling you that these mutations, like the variants you've been learning about, the variants that have been like annotated so often now and being annotated and are categorized as variants of concern. So why this is a way, why there is the variant of concern? Because it concerns because it has such a severity compared to the previous version of the virus, right? So a mutations, I mean, how much they affect, and it's fascinating that simply like the virus mutates and makes it itself such a devastating, I mean, cause such a devastating effect. So I'm really excited to learn about what is the mechanism with which it does that. So how do you know about the mechanism? I mean, you know about the mechanism when you have the knowledge of how the sequences, what sequences it has, what changes it has, made to do this, and then see that in the protein structure. I mean, what is actually happening? So why it is? I mean, what has changed? And if you can compare those two structures, then you'll gain a lot more insights about I mean, maybe how it affects the function, and then because of that it affects all of that happens as a downstream effect, right? And then we have the review session. So this is the last session of the week, and this will be through this these sessions will be at 10 a.m. 10 a.m. time, Eastern time. And for our students and participants in India, it will be at 7:30 p.m. Indian Standard Time. And for people from other time zones, you could, I mean, please, you can convert this. And in this session, it would be about the guest session, and there will be an interactive activities that Dr. Sona has planned for all of you. So in the guest session, we have Dr. Felicia, and I've been like really, really excited for this session. I was when this was confirmed that she'll be able to help you and like give us all a wonderful session about her journey in Uniprot. So she is one of those person, and in this program, both Dr. Sona, as Ilya told, and as you saw, that Dr. Sona was talking a lot more about the history of bioinformatics. So Dr. Celicia here is also one of those people who have been there from the very beginning as a biocurator, and managing the enormous growth of data. So it would be really exciting to hear her about her journey. So that would be the session, last session of that week. And after the session, we have the weekend activity. As I've already told you that with every session, we have a lot of online courses that would be made available to you. So you can learn at your own pace, and you can go through and learn about some of the technologies and try to apply that to real research, because that would be one of the outcomes that we have seen in the program. So in this activity, when you have learned about visualization tools and such as PyMol, then you will be able to apply this into a disease, or finding a structure that has that mutation, which makes it more dangerous, or which makes it to identify those sort of projects. And then we will be using PyMol. So in this weekend, you will have that assignment to prepare a few slides about and find the structure, and find and use PyMol and all of that. So the second week is about research. As I was telling you, it would be from our team at OmicsLogic. And in this session, we will start in this week. We will start from literature review and project design. Here we will learn about the review of what you have learned in the previous session, and we will learn about virology, and we will learn about how you can ask about questions like how big data research, what big data research project is, what is the research question that you're trying to ask, and how you can generate that research question. So as I was discussing before, that this is to design a research project, to be able to work on a research project, and to come up with presentations, and come up with posters or maybe come up with a publication when you have finished the analysis, which would be a very later phase of this entire journey. So this session will be dedicated to those who would want to work on projects. And then we will learn about these viral species. Here we will learn about how genomics is applied in virology. Followed by this, we will learn about how metagenomics is a technique. It's such a powerful technique that you should all be aware of. You should learn about that, how how to apply and understand metagenomics that can study, I mean, directly these samples without culturing in the lab. Very powerful technique, and is being widely used in many of these natural and many of these difficult times in this pandemic. So followed by this, once we have the understanding of genomics and its application, metagenomics and its application, we will learn about the vaccine, antibiotic, and antiviral drug and its strategy. Here we will learn um, about the basic principles behind antiviral and antibacterial drug. As I was telling you that from structure, how you can understand and design these molecules, design these bigger molecules or smaller molecules that could help us actually find solutions, right? So followed by this, we will have the hands-on session. In this hands-on session, we will go to NCBI and we will take the entire genomes and do the analysis through the TBioInfo platform. And in this entire course, you will be also using the TBioInfo platform, which is a very powerful and comprehensive tool, cloud platform, which is an amazing place to do your projects online. So yeah, so in this session, we will be using all of that resources. And towards this end of the week, we will be helping you and work on your projects and come up with these project presentations that could be reviewed by the judge in our symposium in August. So to tell you more about the full program registration and how you can join this program.
Sorry, Mohit, maybe right before you jump into that, could we launch the poll and just kind of learn a little bit more about the participants? That's okay.
Okay.
So
Yes.
Mohit, yeah, you go ahead, Sonanika.
Yes, Ilya. So there is some problem at my end. I'll get to you regarding this. Dr. Mohit can take this session.
No, the
ahead
poll
now.
is on. Sonanika,
is
the
it
poll
on?
is live. Yes, people can see it and their answers. So I'll just guide you, everyone, through the questions that we have.
Yes, maybe.
The first question about your education. So high school, undergraduate or bachelor's, graduate or master's, postgraduate or PhD or postdoc student, researcher or faculty, high school teacher or lecturer or other. So I see that the majority here are undergraduate right now. We'll just give it a little more time. The second question is about your research interest. So what are you most interested in? We chose topics that are somewhat general and broad. So genomics, bioinformatics, data science, the future of medicine, or introduction to omics in general, different types of omics data. So if you could respond to that question number 2. I see about 50% of you have voted now, so please go on. And the final one is, where are you from? So which country are you from? United States, India, Africa, United Kingdom, Sri Lanka, Bangladesh, Nepal, Russia, Turkey, or Iraq. So this was based on some of the submissions that we already had. And so that's why this was inserted. So Abukari, if you could answer in the poll, that would be helpful. Okay, and I think we'll leave the poll on for some time, and Mohit, maybe you could continue and speak a little bit about the registration.
Yeah. Thank you, everyone, for answering the questions on the poll. And if you have any more questions that are not there, could you please unmute yourself and ask me also. I mean, if you want to ask, if you have any questions regarding the program and the sessions and what we would cover, please, you can ask me also by unmuting yourself. About the full program registration, so as you have already gone to the Learn platform and have a Learn portal and have created a login, I think next I will ask Snalika to take you to the program page and show you the registration details. So Snalika, would you be able to do that, or
Yes,
should
sir.
I go
Thank you
through
so
that?
much. Thank you so much, Dr. Mohit. So I hope you all have already signed up on our portal. If you have not, then you can do so right away. I'll wait for 2 minutes for you all to please sign up. I'm sharing the link with you all. So once you are signed up on the OmicsLogic portal, I'll just log in so that you can see how you have to sign up. You need to add your email ID, create a password for yourself and sign in. And you can also link your social and sign up as well. So let me do through my social account so that you get to know how it would be done. You can now sign in yourself on the portal with me. And please tell me if you have done so with a yes or one maybe on the chat box. So once you're signed in on the portal, you need to go to the programs, and here you will get the list of all our programs which are ongoing and the upcoming ones as well. So here you will get the program Genomics in the virtual lab. And as you can see here that you would get all the information for the program on the program page. You would get to know about the program. So this is one of the project videos of our students from the Genomics in the Virtual Lab 2020, the program which was conducted last year. And we had amazing outputs. So I'll be shortly taking you through the blog as well, and I'll tell you where you can find more details about the projects that were conducted by our previous year students. And on the program page, you will have the details of each and every session as well. The dates would be marked on the calendar here, the topics that would be covered in each and every session. And this program page is also to be used for your registration. So those who want to register themselves for the program, they can simply go to the program page and register themselves as well. Now, how do they do so? I'll just quickly take you through it. This is the program page, and you need to start your application. And I would be sharing the details with you of how you would take your application ahead the moment we receive your registration with us. And I had few of the queries regarding certification. So yes, after the completion of the program, you would be getting the certificate, and this completion would be awarded once you finish all your assignments for the program and the tasks given by you by the mentor. And I just told you that we have all the session details as well on the program page and each and every topic that would be covered. So once we cover the session, once the session is over, we would be uploading the video of the session. Now, this is just an example program page. So we have the program ongoing machine learning for biomedical drug discovery. We are done with the 3 sessions. We have the 3rd one today. And the 2 of them, the video has been uploaded here. So we have in the webinar also the people who have already enrolled themselves for the program. So this is for your knowledge as well, that you will get the link to the program page. You need to bookmark it and keep it with you throughout the program. And you would be able to go to the program page and look for the video if you missed out anything. And you can refer to the video anytime and do your assignments as well. And also you would have the Zoom session links for the upcoming sessions, and you can simply log in to the session whenever the date of the session is scheduled for. And now let me take you through few of the more resources that you would get the access to along with the program. So by being a part of the program, first of all, you would also have the access to all the courses on our portal. And also you would definitely have the access to the T-Bioinfo server. Now, this is a very powerful server. Now, why do I call it powerful? It's just that because we have different sorts of utilities over here, which have got integrated pre-built libraries, which could be used to carry out different sorts of tasks and analyze your data sets. And we would get into more detail of it in the program itself. And to get more details on the program about what all would be covered, who is the speaker, and you can just go to our blog page. Now, this is the blog for the program, which has all the details of the program. And talking about Nishchay is a collaborator from Sri Lanka. And then we have the session details here as well on the blog, which you could refer any time. And as Dr. Mohit has already discussed all these sessions, they are mentioned on the blog as well. I would also request you all to please follow us on social so that you stay updated with all our upcoming programs and also all the opportunities that we have. And also we keep on posting a lot many opportunities in the field of bioinformatics, about internships, about recruitments, and a lot many more things as well. So being a part of this group on LinkedIn would definitely help you. So I've shared the link with you. You can join our group. And also we are there on Facebook by the name Pine Biotech. And again, I'm sharing a link with you all. Join us there as well so that we stay connected and you don't miss out on any of the opportunity and the program that we would be having. And before I finish sharing my screen, I would want to take the question. So Anik asks,
okay,
how
thank
much
you,
do we
Sonalika.
need?
If I could
Yes.
interrupt you just for 1 second.
Yes.
So let's go through the poll results just to kind of share with all of you what that looks like. It looks like we have the majority are undergraduate students, and the second largest category is graduate, masters. So the reason I'm starting with this question is because we do have a number of additional programs that are targeted to the more advanced students, including a research fellowship where we're just exclusively focused on helping you complete your research using bioinformatics tools. And there we have a number of mentors that you can interact with that are working using bioinformatics in a variety of different research fields. So on the same program, you will find on this page, you can see in the top menu, there's a link to research. So Sonalika, if you could show the research page. So if you are a graduate student or an undergraduate student or a postgraduate student or even a researcher that would like to work on a research project, we have this 3 or 6 months research fellowship program. You could check that out. Now let's go to the next. What is your research interest? So a lot of you here are interested in genomics, and as we already mentioned, genomics has many different applications. And Sonalika, if you could now go to the home page. On the home page, we have the 4 categories of different types of projects that we have. Yes, the home page. Okay, so if you scroll down, we have here, there we go, oncology, infectious diseases, neuroscience, and agriculture. So what you will find on this portal are not just lessons and tutorials, but also project examples. And the project examples are sourced from the public domain, so they're from journals. Please go back, Solenika, just stay on the 4 sections, please. So in each one of these categories, we have project examples from peer-reviewed journals. We take the data, follow the same analysis approaches that are described, provide some insight into alternative methods of analysis, and also open it up for your independent project on the same topic. And so now, Sonanika, if you could go down below and show how some of the research fellows that we've had in the past have been able to use this training to design a project of their own. So we have a whole page dedicated to this, and you can see here a few of the examples. So if you would like to work on an independent research project, the Genomics in the Virtual Lab program is a great place to start. And as you get started, you will see how we have a whole structure for you to follow, from a basic research question to identifying the data set to then being able to work on an independent project. Okay, and then finally, we did ask about what country you're from. So we have here India is the largest. The second one was from Africa, and then the third one is from the United States. So from India, I did want to mention that we have a number of campus ambassadors that you can connect with if you have an interest in finding out a little bit more about how this works, learning about the research fellowship program, if you have any questions. So we have a whole set of undergraduate and graduate students in our research fellowship. And so we invite you to connect with them. And the way to connect with them and maybe even hear from their experience is to email Sonalika or join our social media group on LinkedIn and on Facebook, and letting us know that you would like to talk to someone. So we will be sharing with you some details, and yes, you can email marketing@pine.bio if you have any other questions. Thank you. Okay, and so we will, I saw that there's a question, so Nika, maybe you can just show, if you log out from your Learn account and show the program page without being logged in, and just walk us through the registration pricing and what are the different options.
Thank you, Ilya. And by the time my page is loading, I just wanted to tell that we are also there on Telegram and WhatsApp. So we have groups for Telegram and WhatsApp as well, so that we have direct communication with you all. And I would be sharing the links of our groups as well. And you can be a part of our groups and ask us anything, anytime, and we would be there to help you out with that. So for the registration process, here on the program page itself, you go to the same link and then you need to start your application. Here is the link. You need to start your application and you would be directed towards the different sorts of level we have for this program. So we have the beginners level, the intermediate and the advanced level. And the difference is just because of the different sorts of provisions that would be given throughout the program. So the beginner one has got the provisions mentioned, and intermediate one has got one-on-one mentor support as an additional benefit as being part of the intermediate level for the program. And the advanced level would have some more options, as in to get extra time to work on a project. And then the option is there for certificate for completion. That is though for every level that you would be in, but there is also an option for certificate of excellence, as you would be getting some more time to work on a project of your own, right? And to talk about the further details, so let's say any one of you chooses the intermediate level. Let's say you just need to sign in and then check out. So the moment you check out, you would be directed towards the payment page. And once you're directed towards the payment page, you need to add the coupon code that is shared with all of you over the mail IDs. And sorry, and if you didn't get one, then you can let me know and I will share with you once again. Sorry, my system has got, yes, it's working now. Yeah, so that's it. I hope your doubt is cleared, Anik. So anybody else, if you have any doubt, you can ask. I'm till then sharing the links of ours. So Kia asked, the price is not affordable for us Africans in Ethiopia. So Kia, we would be sending, can you please drop me a mail at marketing@pine.bio so that we can discuss about the different sorts of opportunities. Anushka says, can you please share the code? Yes, sure. Anushka, can you please do the same as I asked Kia to do so? Please mail me and I would be able to revert you back just within a few minutes. And I'm sharing the link of a Telegram group. So be a part of a group. We have a channel and also a group by a name of OMICS. That's a bioinformatics community where we have students of bioinformatics background and different fields as well. And there we have free live sessions every week, and we discuss different sorts of topics. Like we had one session yesterday where we discussed about astrobiology, and our research fellows were there, Shubhrajit and Dilara Dekan, who shared about astrobiology and how it could change the whole way, the way we look at science. And it was really interesting. So if you guys are not following us on YouTube, you can do that right away. I'll share the link of a YouTube channel as well, because we keep on uploading videos for each of our session there. And also we are live right now, by the way. And being a part of a YouTube community, you would definitely be able to look at all the webinars and sessions if you miss out any one of them. And yes, you could be benefited in that way. So this is our YouTube channel. It's link. Okay, Dr. Pankaj asks the feedback link for participation certificate. Sorry, Dr. Pankaj, feedback link as in feedback link for the program, for the certification feedback link, I didn't get, sorry. Could you please specify what you are saying? Abukari has a question. He says working with, yes, sir.
Let me answer that one, please.
Yes.
So Abukari, we have some tools that you can use with amplicon sequencing for Plasmodium falciparum. And maybe what I would suggest, depending on your level of bioinformatics, your previous bioinformatics, let me ask you maybe a few questions so that we can understand. Do you already have experience using bioinformatics tools for genomic data analysis, Abukari? You can also unmute yourself, I think. Let me actually find you so I can help that. So the tools that we have. Okay, yes, and I think, Sonarika, for you, it's saying that the WhatsApp group is already full, so you can't join that.
One more link, please.
Okay, so Abukari, I've asked to unmute you if you can.
Okay, can you hear me?
Yes, we can hear you.
Good. So I started with bio, infectious disease, and we just completed the course yesterday. I also did start with transcriptomics. I'm just left with some few courses to finish, and this is how far I have started with bio. I hope to register this particular course, and my interest in bio basically stems with the fact that I want it to help me impact positively in my upcoming project that we're going to do amplicon sequencing with PHYLOGENY.
Okay, so because you've already done a lot of the training and you probably have a theoretical understanding of what are the steps that you need to do to get you to now apply these skills that you've acquired to a practical project, I would recommend joining the research fellowship. So the research fellowship program is intended to help people like yourself that have completed the training go through identifying the type of data which you have, the amplicon sequencing. The application would be to Plasmodium, And then potentially thinking about what kinds of questions would you like to study, would you like to address with your project. Potentially you're looking for vaccine efficacy, or maybe you're looking for drug resistance, or maybe you have some other questions, maybe genetic variation in Nigeria, let's say, or something like that. And so a lot of these topics are best when you do literature review. You can refine your question, understand where you should focus your efforts. Then once you've got the data, you would do the exploratory data analysis and understand the variation. And then ultimately, once you are done preparing that project and sharing it with the rest of the research fellows. So we actually have a whole group focused on Plasmodium. We have a group in Ghana out of the Noguchi Memorial Institute for Biomedical Research. And also we have a group in Africa and Nigeria where they have a few people joined in the research fellowship, and they have this common interest in malaria. So maybe that would be the best next step for your situation, Abukari.
Okay. I'm very grateful. I'm very grateful. Thank you.
All right. Yes, thank you for the question as well.
Okay.
So if anyone else has any question, please go ahead. I think I've made it possible for anyone to unmute themselves. So if you have a question, please do let us know. You can also unmute yourself and speak. And I think the question about the feedback link and the certification, so we don't provide webinar certification. These are really webinars to facilitate discussion and introduce some of the upcoming opportunities. We have quite a number of free webinars that you can join throughout the month. Some of them are related to our research fellows presenting their research projects. Some of them are to discuss important topics like the Omics Club. Some of them are to introduce some of the upcoming training opportunities. So please take a look at either the social media groups or the YouTube channel where we share some of those sessions with everyone. But because we're sharing it publicly, we don't really provide a certificate with those. So that's just to answer that question. Okay, Gerald is asking, what are the bioinformatic tools that the program requires the learners to use? So Dr. Vasudevan comes from a structural biology background, so she will be talking about tools like PyMol, but she also has a lot of experience with genomics, so probably sequence alignment, so Clustal, BLAST, and things like that. As Sonalika already mentioned, you will have access to the TBioInfo platform. So let me just briefly share with you some of the tools that are available. All of you that have registered on the Learn portal will have access to a demo pipeline already for free. So you can go ahead and explore this platform and see what kind of tools are available here. So thank you, Sonanika, for sharing that link. So as you can tell, there are many different tools available, and they're organized in such a way that you can build fairly complex pipelines without minimal background in bioinformatics. There are some examples. You can take a look here at some of the examples in transcriptomics, or here you can see some examples for other tools like phylogenetic tree of different types of methods that we have included. So really, this is many different tools for multiple sequence alignment, for mapping of the raw reads to the reference genome, of building phylogenetic trees. There are tools for processing data and analyzing it with different types of unsupervised and supervised methods of analysis. So those are the tools that are going to be covered in the program. But what you should probably know coming into the program are some of the basics about sequences, what they look like, as well as what kinds of questions could you ask. Now, let me share just one more thing. On the Learn portal, let me just go to the home page again, under coursework. A lot of the tools that you've seen on the Tauber portal are actually scripts in R and Python. And so one of the things that you will be able to do is take some of these and learn about how they are implemented. So if you go to the courses, you can take a look at Introduction to Genomics, which is a good place to start, but also you can see getting started with bioinformatics in Python and getting started with bioinformatics in R. So here you will find some scripts that you can run directly in the browser and understand how to analyze sequences and visualize data. Okay, so what is the role of bioinformatics in discovery of new COVID vaccines? Well, so it's obvious that vaccines are going to have to continue and improve as we see new variants of concern and as we see other viruses coming out and becoming potentially pandemic. So I think there's definitely a huge need for new vaccines and vaccine improvement. Now, as we think about the vaccine function and how they are designed, you will understand the relationship between sequence and structure and then immune response. And in that process, bioinformatics plays a huge role because we need to understand what variants to focus on, what impact do those variants have on immunogenicity, how do they interact with the different receptors. And so that's where the vaccine design field, I think, is going to go, because the process of going from a known sequence and structure to being able to produce those proteins or RNA really is, I think, something that was proven to be very effective in this pandemic. Gerald is asking, is the program geared towards structural biology and biophysical methods in the context of bioinformatics? No, it's going to combine genomics with structural biology. So how do you do sequence alignment? How do you understand the relationship between sequence and phenotype? And then how can you map this on the structure to understand the relationship between the two? Okay. Okay, thank you. So I don't see any more questions. So hopefully we were able to answer all of those.
Thank you so much, Ilya. And the last thing I wanted to share with all of you is that we also have a lot of programs which would be starting by the end of June and also in the first week and mid of July. So we had a question regarding SARS-CoV-2. So we have this program coming up, which would start from 9th of July, and you can just go to the program page and get the details of it. And if this interests you, maybe you can be a part of it and learn more about how we can use bioinformatics effectively to do research in this particular domain. And then we have bioinformatics for infectious diseases program as well. That would be starting on 22nd of June. And last but not the least, we have the genomics in the virtual lab, about which we are here to discuss about. And the program would be starting on June 21st. And the last day to register for the program is June 20th. We would be closing the registrations at the end of June 20th. So you all are requested to register, if so, for the program. And yes, that's it. So thank you everyone for joining today's webinar. And if you have any doubts and if any of your question went unanswered or you could not ask, you can anytime mail us at marketing@pine.bio and mohit@pine.bio. And me and Dr. Mohit would be there to help you out at any step. India, Dr. Pankaj has a question regarding clinical trials of vaccine, whether it is helpful. Can you please take that?
I would just maybe suggest we ask a few questions. So Dr. Pankaj Kumar, maybe we could schedule even a conversation. It sounds like maybe you're involved in related research, so we'd love to learn a little bit more about it. You're welcome to email me, ilya@pine.bio. And I'd love to set up a chat to learn more about your interest.
Thank you, Ilya. And Dr. Deepak, we would definitely look forward to have a conversation with you. And Gerald, you can't log in into Tau service like that. So you need to first sign up in the OmicsLogic, and then you need to be a part of our program, any of the programs that we have. It could be the program for which we are here today, or the research fellowship program, or the programs that would be coming up, and then you would get the access to the BIOINFO server. We would be giving you the access for that. And if you want the access to the server to carry out your research work only, and then you can reach out to me for that. I'll send you out the details how you could get that, and then you can maybe use the server with the credentials and do your research. I hope that answers your question. Animika says you want to develop your project on neuroscience, what options I have. So Animika, we are already working on a lot of new neuroscience projects in our research fellowship program. I received your message over LinkedIn as well. I'll send you the details of the program, and I'll also send you the project samples for neuroscience. You can go through them and get an idea of how the research fellows, our ex-research fellows, worked on it. And then we can proceed further. Yes. Thank you so much, everyone, for being here for the webinar. It was really, really great to know you all. And we look forward to your participation in the program and also meeting you on different sorts of other webinars. And stay connected with us over social so that we keep on meeting again and you don't miss any of the update on the upcoming programs.
Sonalika, I have one question.
Yes, please. It's
Yeah.
Durba, right?
Yes, yeah.
Yes,
well,
yes.
I try to click on the, you show the 4 different real project on homepage. Yeah, when I
Yes.
try to click the learn more, I couldn't do that. Do I need to do anything or?
If you're not able to do that, then let me share with you the blog of the research fellowship program, and you
Well,
can just go
maybe
to
I could
this.
just one
Yes.
second clarify. Yeah, so that is a blog post that you should take a look at. But if you go into the courses tab, you'll see that you can search there what is available. So let me just show right here. So if you go to the courses tab,
Yes.
right here, you can search for a specific project if it's available. And so we have a few examples. Or if you would like to develop an independent project in one of these areas, again, take a look at some of the examples that Sonalika shared in the blog. Those are examples of projects that students have completed in the research fellowship. So if you would like to develop something like that, you can go to research, and that tells you a lot of the information that you need to know of how this works and some of the examples down here as well.
Yes, I'd like to develop independent project. Yeah. So probably
Yeah,
join.
for that, for an independent project, maybe we could also schedule a time to speak, learn a little bit more about your background, your interest, what exactly you're interested in. So if you could email Sonalika and myself, and we could schedule a time to meet with you.
Okay, that will be great.
All right.
Thank you so much, Durba. I hope that helps you. And thank you so much, everyone, for being a part of the webinar. Let's meet the next time. Thank you so much. Bye-bye. Take care.
Okay, bye-bye.